My sincere thanks to ToxMaster Class India. Also, my pranams to my gurus both in toxicology and pathology. It's really an honor and privilege for me to give this presentation. Lot of information is covered. Use this as a scaffold and do further reading to deepen the understanding on toxic responses of the liver. So in the first part, I will be covering these topics: liver cells and functions, bile formation, liver pathophysiology, fatty liver, endothelial cell damage, inflammation, regeneration and repair, fibrosis, liver cancer, classification of liver injury, histopathology and clinical chemistry, intrinsic versus idiosyncratic response, and initiation and progression of hepatotoxicity. In my next talk, I'll be covering the rest of the things which will be on July 20th. So, toxic responses of the liver. Let us look at the liver performs many functions that are critical to life. Liver functions are complex and diverse with exocrine and endocrine activity. And liver is also involved in metabolism, conjugation, detoxification, and hematopoiesis in early embryonic stages. So, the blood supply to liver is very unusual. Arterial blood supply is there. It is it's minor. That is 1/3 in human beings and lesser in rodents. Whereas portal blood supply is major, and it's 2/3 in humans. Hepatic portal vein is the venous drainage from stomach and intestine, so oxygen is less. Also, all foodborne xenobiotics absorbed from GI tract are brought to the liver first. So this is the same thing what I explained. This is a schematic representation. The portal vein comes from the gut and also splenic vein joins, and that's the portal vein. Aorta gives rise to hepatic artery. That's the hepatic artery entering the liver portal. And in the liver, the veins, all the veins form together hepatic vein and then join the inferior vena cava. So this is the structure of the hepatic lobule, and the liver is made up of hepatocytes, and there is a portal vein, portal triad, which is comprising of portal vein, hepatic artery, and bile duct. So you can see sinusoidal spaces are there. The blood flow is from this sinusoidal space and they go to the central vein. From the central vein, it will be forming the hepatic vein and they join the inferior vena cava. So this is the structural unit, whereas this is the functional unit. The functional unit of the liver, as you can, the hepatic, this hexagonal structure is divided into acinar. When the functional is, it starts from the, see, this is the portal area. And then if you can, the division is zone 1, zone 2 and zone 3 towards from the portal vein towards the central vein. So when the zone 1 in the periportal, mid zone is the next to that, and the zone next to the central lobular is the zone 3. So there are differences in functions that exist depending on where the hepatocytes are located along the sinusoids. Zone 1 is maximal bile acids from plasma are extracted in zone 1 hepatocytes compared to zone 3. That is physiological concentration. Then in zone 1 is rich in mitochondria. They predominate fatty acid oxidation, gluconeogenesis, ammonia detoxification to urea. And higher concentration of glutathione, that is GSH, is there in zone 1. Whereas zone 3 is rich in cytochrome P450 enzymes. Liver is important for homeostasis. Damage to hepatocytes and other liver cells leads to liver dysfunction and also dysfunction of the other organs. That is, for example, hepatic encephalopathy, coagulopathies. So this cartoon, I'll be using it many more times, but this is to highlight these are the hepatocytes. So next I'll be talking about the hepatocytes. Hepatocytes, they are the hepatic parenchymal cells. They are large epithelial cells. They process food. Monosaccharides to glycogen is synthesized. Gluconeogenesis happens here. Lipids processing energy. Then synthesis of circulating lipids. Then uptake of dietary lipids, cholesterol, and vitamins from blood. degradation of cholesterol and steroids. Protein synthesis for intrinsic and extrinsic protein, albumins, coagulations, and complement factors, lipoproteins. Then ammonia detoxification, heme synthesis, elimination of bilirubin, iron reutilization, xenobiotic metabolism, drugs, foodborne agents, etc. Excretion via biliary tract, drugs and metals, elimination of particulates and bacterial products from blood. These are the hepatocytes functions. Now, we will look at the sinusoidal space. This is the sinusoidal space, which is in the center, which takes the blood from the portal vein to the central vein. So, in that, and there is a space of Deci, that is the interstitial space. This space is between the endothelial cells and the hepatocytes. And these cells, you can see that elongated cells which are lining the sinusoidal space, they are called the endothelial cells. They have the fenestrae. So let's look at the endothelial cells. What are their functions? So sinusoidal endothelial cells, these sinusoids are lined by discontinuous endothelial cells which are having fenestrae, that is pores. Fenestrae are pores which allow molecules less than 250 kilodaltons to pass through to cross the interstitial space of Deci. This includes albumin. Interstitial space is the space between the endothelium and hepatocytes. Endothelial cells are important in scavenging lipoprotein, denatured proteins, advanced glycation and products. Glycation end products. Lipoproteins via the apolipoprotein E receptor, denatured protein and advanced glycation products via scavenging receptors. The endothelial cells secrete biologically active molecules such as cytokines, prostanoids, nitric oxides, endothelins. Intercellular adenosine molecules ICAM1 and vascular cell adenosine molecule VCAM1 are secreted on cell surface. They are important for injury and repair. So coming to other cells, there's something called Kupffer cells and stellate cells, dendritic cells. Kupffer cells are the macrophages. They are, just notice that it is connected to the endothelial cells. They are in the sinusoidal space, and in the space of this interstitial space, you can see the stellate cells. I'll come to that, what their function is. Just understand this. And there are dendritic cells. They are also in the interstitial space of Deci. So let's look at Kupffer cells. These are resident macrophages in liver. They are situated within the sinusoids and their processes reach the hepatocytes through the fenestrae of sinusoidal endothelial cells. Kupffer cells ingest and degrade particulate matter, especially bacteria. Major source of immune substances like cytokines, eicosanoids, and reactive oxygen species, and can act as antigen presenting cells. Then coming to hepatic stellate cells, they are also known as eto cells and fat storing cells. These are very important cells. They have two roles. Located between endothelial cells and hepatocytes. They are vitamin storage sites. High dose of vitamin A therapy leads to engorgements, proliferation, and protrusion of stellate cells into the sinusoids. They also have contractile function. Cells secrete smooth muscle actin, which give them a contractile properties. The role in liver injury and fibrosis. In chronic liver injury, these stellate cells can transform to myofibroblastic phenotype, synthesizing and secreting collagen, extracellular matrix protein, and thereby initiate fibrosis. So, to compile the hepatic stellate cells, they are located between endothelial cells and hepatocytes in liver sinusoids. Other names, eto cells and fat storing cells. Primary function is storage of vitamin A. Contractility, they express smooth muscle actin, regulate sinusoidal blood flow. Role in disease, activate during chronic injury and produce collagen, leading to fibrosis. Other immune cells of liver sinusoids. Immune cells in liver sinusoids are strategically organized to maintain homeostasis and respond to injury or disease. These cells can become activated by inflammatory stimuli and contribute to aid in liver injury or tissue repair. Key immune cells beyond Kupffer cells are dendritic cells, plasmacytoid dendritic cells. Both these cells play a key role in antigen processing and presenting antigen to other immune cells, and they also produce inflammatory cytokines like interferon. Dendritic cells have protective role. Their resolution of fibrosis and acetaminophen hepatotoxicity. So natural killer cells and natural killer T cells, I have just tabulated this so that you can get the final summary of it. Natural killer cells, they are they have primary role in inflammatory and antifibrotic. The notable function is cytokine production, killing of activated stellate cells. There other points is mixed evidence is there in acetaminophen toxicity. Natural killer T cells, they are pro-inflammatory in some contexts. Example, alcohol injury. Cytokine production and modulation of immune responses are notable functions, and mixed evidence in acetaminophen toxicity. Innate lymphoid cells, ILCs, they respond to injury-induced signals, that is IL-33. They promote fibrosis and inflammation. This is the emerging area of research. Now let's go to bile formation. Bile. Bile is yellowish colored fluid produced by hepatocytes and plays a crucial role in digestion and detoxification. The composition of bile is water, which is a main solvent. Bile acids facilitate lipid digestion and absorption. Glutathione, that is GSH, acts as an antioxidant protecting intestinal cells. Phospholipids and cholesterol, they help solubilize lipids. And bilirubin, this is a breakdown product of hemoglobin responsible for bile's color. Organic anions, proteins, metals, ions, xenobiotics involved in detoxification and transport. Importance of bile formation, lipid nutrient uptake. Bile acids emulsify fats in the small intestine, enabling their absorption. So protection against oxidative damage. GSH in bile protects the intestinal lining. Excretion, especially in removing the endogenous waste products and xenobiotic compounds from the body. Mechanism of bile formation and secretion. The role of hepatocyte in canaliculi is hepatocytes. These are the specialized cells which they help in the bile production. Canaliculi is a small channel formed by the plasma membranes of adjacent hepatocytes where bile is secreted. I'll be showing that soon, that diagram. These canaliculi are sealed off from the intercellular space by tight junction. The tight junction and selective permeability. Tight junctions allow passage of water, electrolytes, and some small molecules. They are impermeable to organic anions, which permits the accumulation of high concentration of bile acids, GSH, bilirubin-like diglucuronide, and other organic anions in bile. So these are the bile canaliculi. These are small channels and in adjacent to two hepatocytes, their channels formed. So bile is secreted into these bile canaliculi. So let's look at the process. The biliary tract resembles a tree. Like how? Canaliculi, they are the root tips, connect to intrahepatic bile ducts. They are larger channels within the liver. These ducts merge into extrahepatic bile ducts. The extrahepatic ducts combine to form the common bile duct. Then there are this gallbladder. Gallbladder stores and concentrates bile before releasing it to the duodenum. That gallbladder is absent in some species, example horse, whale, and rat, where bile flows directly from the common bile duct to the duodenum. So let's look at the bile formation and transport. This is a complex process and I have simplified and I have put it. Please read it thoroughly and you will get it. It's not as complex as it is seeming, but there are a lot of transporters involved. Then you need to know about this for especially for toxicology. When it is inhibited, what happens? Okay, so hepatocytes initiate bile formation by two processes: by actively transporting bile acids, that is, and glutathione, and other osmotically active compounds, including xenobiotics and their metabolites, into the canalicular lumen. This is the canalicular lumen, and that is called bile salt dependent flow. And that happens by the transporters, which are seen here. And by using different that transporters method, they are secreted here. And there is one more method. This active transport creates an osmotic gradient that drives the passive movement of water and electrolytes across the hepatocyte epithelium into bile. This process is known as bile salt independent bile flow. So water and other electrolytes are also drawn into the canaliculi. So, four steps are involved: uptake of bile acids and other solutes into hepatocytes, intracellular transport and metabolism, canalicular transport, apical transport into bile, bile duct modification, absorption and secretion of solutes by cholangiocytes. So, it's everything is summed up in this slide. So, from the blood vessel, bile acids and other solutes are taken into the hepatocytes. How they are taken is there are two different methods, that is sodium independent uptake and sodium dependent pump. These are all using the different transporters. OATP is a transporter, NTCP is a transporter, MRP is a efflux pump, whereas OCT is a transporter through which all these bile acids and other solutes are taken into the hepatocytes. In hepatocytes, the bile acids and solutes bind to cytosolic protein like 3-alpha HSD, hydroxysteroid dehydrogenase, and liver fatty acid binding protein for intracellular trafficking towards the canalicular membrane. So they go towards the canaliculi and they again transported to canaliculi through again some transporters. This is MDR3, ABCG5, ABCG8, MRP2, and BSEP. BSEP are very important. You must know bile salt export pumps. Through this, they are exported into the canaliculi. And when the canaliculi merge and they go into the bile duct, here the cholangiocytes, they also return some of the conjugated bile acids to the portal vein. Some shunt keeps happening there. So this is in summary, in a nutshell, how the bile is formed. Now let's look at liver pathophysiology, types of liver responses to chemicals. So I've put the various liver responses and representative toxicants. Let's look at the various responses. Hepatocyte death, fatty liver steatosis, canalicular cholestasis, bile duct damage, sinusoidal endothelial damage, inflammation, liver regeneration, fibrosis and cirrhosis, and liver cancers. The various toxicants are majorly it is acetaminophen for cell death, amiodarone for fatty liver, chlorpromazine, canalicular cholestasis, alpha-naphtha isocyanate for bile duct damage, cyclophosphamide for sinusoidal endothelial injury, bacterial endotoxin, inflammation, and ethanol inflammation, liver regeneration, acetaminophen again, estrogen again, fibrosis and cirrhosis, carbon tetrachloride, ethanol, thiostemide, vitamin A, vinyl chloride, liver cancer, aflatoxin, androgens, etc. So let's look at each one by one. Okay, let's look at cell death. Cell death from chemical exposure occurs via multiple molecular pathways leading to different modes of cell death such as oncotic necrosis, apoptosis, pyroptosis, and necroptosis. There are two new terms introduced here. You all must be familiar with necrosis and now apoptosis also. The necrosis, oncotic necrosis, often simply called as necrosis. It is characterized by cell swelling, leakage of cellular content, nuclear disintegration, karyolysis, and an influx of inflammatory cells. Lot of inflammatory cells will be there. Cellular contents released include enzymes like alanine aminotransferase, aspartate aminotransferase, which enter the plasma and serve as biomarkers of liver cell injury. Necrosis leads to inflammation due to release of intracellular contents into the surrounding tissue. Now let's look at the other one, that is apoptosis. Morphologically distinct from necrosis, apoptosis features cell shrinkage, whereas the necrosis cell lysis, swelling and lysis, here it is shrinkage. Then there is chromatin condensation, nuclear fragmentation, membrane blebbing, and formation of membrane bound apoptotic bodies. Apoptotic bodies are phagocytized and digested by Kupffer cells or neighboring cells, typically without triggering inflammation. Apoptosis is mediated by caspase enzymes that cleave nuclear DNA and other proteins critical for cell survival. The two main pathways that trigger apoptosis are extrinsic pathway, another is intrinsic pathway. Extrinsic pathway, death ligands like Fas ligand, TNF alpha bind death receptors, forming a death inducing signaling complex that activates initiator caspases, that is caspase 8 and 10. This leads to mitochondrial involvement where proteins like truncated BID and BAX promote cytochrome C release, forming the epitosome and activating effector caspases, that is caspase 3 and 7 that execute apoptosis. Then there is intrinsic pathway. Intrinsic pathway is triggered by cytotoxic stress or DNA damage activating P53, which promotes pro-apoptotic BCL2 family proteins, leading to mitochondrial release of cytochrome C, SMAC, and other factors activating caspases downstream. Apoptosis in liver cells involves mitochondrial signaling, but strong Fas receptors activation can bypass mitochondria. So, apoptosis is generally non-inflammatory due to containment of cellular contents and rapid clearance by phagocytes. There is new upcoming one more terminology, that is necroptosis, a form of programmed necrosis initiated by death receptors like TNF receptor 1. Involves formation of complex including receptor interacting protein kinases, that is RIP1 and RIP3. If caspase 8 is inhibited, RIP1 and RIP3 activate, that MLKL, which translocates to the membrane to form pores, causing cell death, independent of caspases. So necroptosis shares features with necrosis, but it is regulated process. Its role in liver disease models is still under investigation, somewhat controversial. Now, additional notes about cell death. Different modes of cell death can share molecular features, especially mitochondrial involvement, complicating clear classification. Histological examination remains the gold standard for distinguishing cell death types. Apoptosis is characterized by anti-inflammatory outcomes, unlike necrosis, which promotes inflammation due to cell lysis. Understanding these pathways is critical for interpreting liver injury from toxicants like acetaminophen and others. In summary, chemical exposure induces hepatocyte death primarily through oncotic necrosis and apoptosis, each with distinct morphological and molecular features. Necroptosis, a special form of cell death, represents a regulated necrotic pathway with emerging significance. The interplay of these pathways underlies the complexity of liver injury mechanisms. So these are the two images of liver. The one on the left is the one which is showing the arrowheads are showing the necrosis. Their cells are swollen and undergoing necrosis, and they're disintegrating and they're lysing. Okay, whereas this arrow is showing the apoptotic cells, they are shrinking and they are undergoing karyorrhexis and karyolysis. Okay, this is pale eosinophil, this is dark. Whereas in this image on the right side, this is the overdose of acetaminophen and the toxicity. Around the central vein, the liver cells are dead. So the necrotic liver cells. What is missing in the photo is that it doesn't have the inflammatory cells. I think it's taken before that. Now let's go to the fatty liver. Fatty liver or steatosis is defined as an appreciable increase in lipid content, mainly triglycerides, within hepatocytes. Steatosis is classified based on the size of fat droplets into macrovesicular steatosis and microvesicular steatosis. Macrovesicular, as the name indicates, they are characterized by one or more large fat droplets that push the liver, that push the nucleus to the eccentric position of the hepatocyte, like signet ring. This is the most common form of typically associated with conditions like alcohol use, obesity, and diabetes. Whereas microvesicular steatosis is characterized by many small lipid droplets filling the cytoplasm without displacing the nucleus, which remains central. This form is less common and may be seen in specific severe liver diseases such as acute fatty liver of pregnancy and Reye's syndrome. The pathogenesis involves an imbalance in fatty acid metabolism. Free fatty acids originate from hepatic synthesis, mainly from carbohydrates derived acetyl coenzyme A, hydrolysis of absorbed fat, and release from adipose tissues. These free fatty acids can be either oxidized in mitochondria via beta oxidation or esterified into triglycerides for storage or export as very low density lipoproteins. Disruption in this balance, such as increased free fatty acid intake, decreased beta oxidation due to mitochondrial dysfunction or enzyme inhibition, or impaired VLDL secretion, leads to the triglyceride accumulation and steatosis. Protein synthesis inhibitors and certain drugs can cause steatosis by inhibiting the protein compounds required for VLDL assembly and then secretion, or by impairing the mitochondrial beta oxidation. Examples include valproic acid, amiodarone, tamoxifen, and others. Ethanol is a major steatosis inducing agent causing mitochondrial damage and increased lipid accumulation. Steatosis is generally reversible if causative stimulus is removed. However, persistent steatosis increases susceptibility to further liver injury, such as ischemia, and can progress to steatohepatitis, fibrosis, cirrhosis, and hepatocellular carcinoma. This image, you can see lipid accumulation. They are macrovesicular, they are all big, and in liver section stained with oil red O. Nuclei are stained blue. So in summary, fatty liver is marked by triglyceride accumulation in hepatocytes, visible as vacuoles that displace the nucleus in macrovesicular steatosis. Macrovesicular steatosis features large lipid droplets with nuclear displacement. Microvesicular steatosis shows small droplets without nuclear displacement. Free fatty acids from hepatic synthesis, diet, and adipose tissues contribute to steatosis when their metabolism or export is impaired. Various drugs and toxins can induce steatosis by disrupting lipid metabolism. Persistent steatosis can lead to progressive liver disease. So let's continue. The next is canalicular cholestasis. Cholestasis is characterized by elevated serum concentration of compounds normally concentrated in bile, particularly bile salts and bilirubin. This liver dysfunction is defined as decreased in the rate of bile formation or impaired secretion of specific solutes to bile. When the excretion of bilirubin is impaired, it accumulates in the skin and eyes, causing jaundice. Excess bilirubin is excreted in urine, darkening its color. Histologically, cholestasis may show dilation of bile canaliculi and bile plugs in bile ducts and canaliculi. Toxicant-induced cholestasis can be transient or chronic and may be associated with cell swelling, death, and inflammation due to intracellular accumulation of chemicals normally secreted into bile, such as drugs, metabolites, and bile acids. Molecularly, cholestasis involves dysfunction in transporter systems on hepatocyte membranes. For example, inhibition of the bile salt export pump (BSEP) by drugs like rifampicin, bosentan, and troglitazone leads to bile acid accumulation and liver injury. So BSEP also plays a major role, remember that. Estrogen and progesterone metabolites can also inhibit BSEP, that is bile salt efflux pumps, after excretion by MRP2. The accumulation of bile acids can cause inflammation and direct cytotoxicity, especially in humans where more hydrophobic bile acids accumulate. So, bile duct damage. Next, let's look at that. Another name for intrahepatic bile duct damage is cholangiodestructive cholestasis. There will be sharp elevation of serum alkaline phosphatase, bile acids, and bilirubin levels like canalicular cholestasis. Histologically, bile duct injury can manifest as infiltration of inflammatory cells in the biliary epithelium and epithelial damage consistent with cholangiodestructive processes. In rare cases, permanent bile duct loss can occur, known as vanishing bile duct syndrome, which has been reported with drugs like antibiotics, anabolic steroids, contraceptive steroids, carbamazepine. So next, let us look at sinusoidal endothelial cell damage. There are four mechanisms. First is dilation or blockade of sinusoidal epithelium, sinusoidal lumen. Dilation occurs when blood flow downstream is obstructed. A rare primary dilation condition called pileosis hepatis is linked to anabolic steroids and danazol. Blockade happens when fenestrae enlarge excessively, trapping red blood cells either within the fenestrae or in the interstitial space of DISI. Second mechanism, destruction or injury of sinusoidal endothelial cells itself. So gaps form between endothelial cells due to direct injury. Example, exposure to acetaminophen, galactosamine, endotoxin, antiphase, antibody, or detachment from the extracellular matrix. Matrix metalloproteinase inhibitors can prevent gap formation. The injury leads to loss of barrier function, causing blood to accumulate in liver tissue. Let's look at inflammation. Inflammation is a protective biological response triggered by injury or infection involving activation of the innate immune system. In the liver, inflammation is a complex process engaging both resident cells and circulating immune cells aimed at removing damaged tissue and initiating repair. However, inflammation can also exacerbate liver injury under certain conditions. Key components of liver inflammation: the resident cells involved, Kupffer cells, liver resident macrophages that play a central role in detecting injury and pathogens. Natural killer, there are NK cells, there are NKT cells contributing to immune defense. There's innate lymphoid cells also, they contribute to the inflammatory milieu. So these are the resident cells there. There are also circulatory cells or inflammatory cells, but which are coming from the circulation. They are platelets, neutrophils, lymphocytes, and monocytes. Mechanisms triggering liver inflammation. There are two different terms I am going to introduce. That is, one is DAMP, another is PAMP. DAMP is damage associated molecular patterns. These are injured hepatocytes release DAMP, such as HMGB1. DAMPs activate receptors like toll-like receptors on immune cells and initiating inflammation. Pathogen-associated molecular patterns, PAMs, they are bacterial products like lipopolysaccharides from the gut can enter the liver via portal circulation. PAMs activate Kupffer cells and other sinusoidal cells through TLRs, again toll-like receptors. Infections in other tissues can also trigger hepatic inflammation. Roles of Kupffer cells. First is Phagocytosis, they remove pathogens and cell debris. Then reactive oxygen species, they generate hydrogen peroxide and other ROS to kill pathogens, which can also cause oxidative stress to neighboring liver cells. There are inflammatory mediators produced by Kupffer cells. They produce cytokines and prostanoids that recirculate neutrophils and amplify inflammation. Recruit neutrophils and amplify inflammation. Anti-inflammatory mediators are there. They secrete prostaglandin E2 and interleukin 10 to downregulate inflammation and promote repair. This the dual role. Kupffer cells can both aggravate liver injury and promote healing by clearing debris. Neutrophils in liver inflammation. They are in activation. They're triggered by bacterial infection or extensive cell injury. Functions, they produce oxidants like hydrochlorous acid via NADPH oxidase and myeloperoxidase pathways. They release proteolytic enzymes like elastase, cathepsins G, and bactericidal protein. The contribution to injury, while essential for defense, neutrophils can damage viable liver cells through oxidative and enzymatic mechanisms. The multi-step process involves adhesions, molecule upregulation, migration out of vasculature, and full activation upon chemotactic signals. So, additional immune cells and inflammatory responses. There are mononuclear cells, especially derived from the blood, also contribute to similar to Kupffer cells, they're monocytes. The other immune cells participate in acute and chronic liver inflammation. The liver's position downstream of the intestine exposes it to microbial products influencing immune responses. In summary, liver inflammation is a double-edged sword, essential for defense and repair, but capable of worsening injury. Kupffer cells and neutrophils are central players with complex roles. The triggers include DAMPs from injured cells and PAMPs from microbial products. Understanding these processes is crucial for developing therapies for liver diseases, including inflammation. Regeneration and repair. The liver has high capacity to restore lost tissue and function by regeneration. Liver regeneration mechanisms after hepatectomy. Liver has a remarkable ability to regenerate lost tissue and restore function primarily through the proliferation of mature hepatic parenchymal cells following significant tissue loss, such as after 70% hepatectomy in rodents or surgical resection in humans. Differences between regeneration after hepatectomy and toxic injury. Hepatectomy triggers regeneration primarily through proliferation of existing mature cells, hepatocytes, without significant necrosis. Toxic injuries, example acetaminophen overdose, require both hepatocytes expansion and also removal of necrotic tissue. Different cell types, particularly macrophages, play distinct roles in repair depending on the injury type. Hepatocytes and cell cycle activation. Hepatocytes are normally quiescent. The mature hepatocytes do not proliferate. To proliferate, hepatocytes must enter the cell cycle, a process initiated by cytokines such as TNF alpha and IL-6. These cytokines prime hepatocytes to respond to essential growth factors like hepatocyte growth factors and transforming growth factor alpha. There are two models of hepatocyte activation: Fasto's priming progression hypothesis. These are here cytokines prime hepatocytes, which then progress through the cell cycle in response to growth factors. Whereas Michaelopoulos model suggests a different mechanism where primary mitogens, the growth factors, directly stimulate hepatocyte proliferation, while secondary substances like TNF alpha enhance this effect. Cell cycle regulation. Cytokines and growth factors activate transcription factors that regulate the expression of cyclins. the activators of dependent kinases, CDKs, then CDK inhibitors, example P18, 21, P27. This coordinated regulation controls progression through cell cycle phases, including DNA synthesis S phase and mitosis M phase. Alternative regeneration pathways. If hepatocytes proliferation is blocked, regeneration relies on hepatic stem cells or oval cells, which can proliferate and differentiate to replace lost liver parenchyma. That's when cirrhosis starts happening. Oval cells are less sensitive to inhibitory cytokine transforming growth factor, allowing them to proliferate when needed. So let's go to liver cirrhosis. Liver cirrhosis. This is a image of liver where there is cirrhosis. Fibrous bridges are there. This is stained blue. This is in cases liver. This is liver parenchyma. You can see the fibrous connective tissue, encasing the liver acinus. So hepatic fibrosis is the excessive accumulation of fibrous connective tissue in liver that occurs as a response to chronic liver injury, overwhelming the organ's repair capacity. It is characterized by increased deposition of fibril forming collagens, mainly type 1 and 3, a decrease in normal plasma membrane collagen type 4. This fibrous tissue accumulates around the central veins, portal tracts, within the space of disease, progressively altering the liver architecture. It can, because they have the ability to shrink the liver, or they go into lobules and they can shrink and make some blebs kind. Causes of hepatic fibrosis: hepatitis B, hepatitis C infection, alcoholic liver diseases, non-alcoholic steatohepatitis, biliary obstruction, autoimmune hepatitis, inherited metabolic diseases like Wilson disease, hemochromatosis, chronic exposure to certain drugs and toxins, heavy metal overload. Sequelae. When fibrous scars subdivide the liver into nodules of regenerating hepatocytes, fibrosis has advanced to cirrhosis, severely limiting liver function. So they form nodules. When they shrink, they form nodules. Pathogenesis of fibrosis: activation of hepatic stellate cells, which I had already mentioned earlier, which migrate to injury sites, proliferate, and differentiate to myofibroblast like cells that produce extracellular matrix protein, including collagens. This activation is stimulated. Reactive oxygen species from injured hepatocytes, pro inflammatory cytokines from Cooper cells, fibrogenic cytokines like transforming growth factor beta 1, platelet derived growth factor. Activated stellate cells also express alpha smooth muscle actin, contributing to increased contractility and portal hypertension during fibrosis. Additionally, the balance between matrix metalloproteinases and their inhibitors shifts, reducing collagen degradation and promoting extracellular matrix accumulation. So, fibrosis is initially a dynamic and potentially reversible wound healing response, but with persistent injury, it progresses to irreversible scarring and cirrhosis, leading to liver dysfunction, portal hypertension, and increased risk of liver failure and hepatocellular carcinoma. In summary, hepatic fibrosis results from chronic liver injury causing excessive collagen deposition, mainly by activated stellate cells, altering liver structure and function, with viral hepatitis, alcohol, and metabolic liver diseases as the leading causes worldwide. Let's look at liver cancers. It must have been covered in chemical carcinogenesis, but anyway, I will give you some salient features of liver cancers. Hepatocellular carcinoma is linked to chronic exposure to chemical carcinogens such as androgens, alcohol, aflatoxin contaminated diets. Additionally, major risk factors of hepatocellular carcinoma are viral hepatitis, especially hepatitis B and C, metabolic diseases including hemochromatosis, alpha 1 antitrypsin deficiency, and non alcoholic hepatocellular steatohepatitis, that is NASH, which is increasing nowadays. There is a well organized synergy between dietary aflatoxin exposure and hepatitis B virus infection, increasing the risk of HCC significantly. Non parenchymal liver cells give rise to other liver cancers. Bile duct cells give rise to cholangiocarcinoma. There's a radioactive thorium dioxide, thorocrast, that is radioactive contrast material exposure that can cause cholangiocarcinoma. The furan also induces cholangiocarcinoma in rats. Kupffer cells holds these thorocasts for a long time and gives rise to gallbladder and bile duct and hepatocyte cancer. Hepatic sinusoidal cells give rise to malignant hemangiosarcoma. There is a occupational exposure of vinyl chloride and arsenic can rise to hemangiosarcoma. The liver micro environment, including fibrosis and immune cell activity, plays a crucial role in hepatocellular carcinoma development and progression. So, I'm coming to classification of liver injury. Liver injury can be classified based on following methodology, each providing a distinct framework and useful experimental and clinical settings. One is morphology, that is histopathology, and serum and plasma biomarkers, clinical chemistry, or the nature of toxic response, that is intrinsic versus idiosyncratic. Histopathology. In experimental animal studies, histopathological evaluation is the gold standard for characterizing liver injury. In humans, liver biopsy is sometimes used, but only under specific conditions due to procedural risk. The histological pattern of acute liver toxicity often follows lobular zonation. For example, in central lobular necrosis around the central vein is common with toxicants to reactive reactive metabolites by cytochrome P450 enzyme concentrated in this zone, zone 3. Examples include acetaminophen, carbon tetrachloride, halothane, pyrazolidine alkaloids. Then there is periportal lesion and the zone 1 occur with toxins like aflatoxin B1 and allyl alcohol. Midzonal lesions predominate with the exposure of bacterial endotoxin. Histopathology. Histologic patterns of drug-induced liver injury (DILI) include acute and chronic hepatitis, acute and chronic cholestasis, cholestatic hepatitis. These patterns correlate variably with clinical outcomes. Histological features assessed include all that which we have studied, which we went through now: inflammation, necrosis, fibrosis, steatosis, cholestasis, and cellular infiltrates. So, no single histologic pattern is specific to drug induced injury. Multiple patterns may coexist, and the same drug may cause different injury patterns in different patients. So, now coming to clinical chemistry, serum or plasma biomarkers. Biomarkers indicate liver function disruption or cellular damage. Conventional biomarkers include enzymes such as alanine aminotransferase, ALT, aspartate aminotransferase, AST, glutamate dehydrogenase, gamma glutamyl transferase, GGT. Other markers include plasma bile acids, total bilirubin, total plasma protein, albumin, triglyceride, coagulation tests. Elevated AST and ALT are common indicators of hepatocellular injury but are not disease specific. The nature of toxic response. Liver injury can be intrinsic, that is predictable and dose dependent, or idiosyncratic, unpredictable, non-dose dependent. Intrinsic injury often involves metabolic activation to toxic metabolites causing centrilobular necrosis. Idiosyncratic reactions may involve immune mechanism such as T cell mediated response triggered by drug protein adducts or mitochondrial injury leading to necrosis or apoptosis. So let's look at that, as a table, characteristic of intrinsic and idiosyncratic hepatotoxicity. In intrinsic, it is with drugs, usually occurs in overdose, whereas idiosyncratic occurs in pharmacological treatment regimens. It is distinctive lesion, intrinsic, whereas in idiosyncratic is variable pathology. It is dose related in intrinsic, whereas in idiosyncrasy it is dose relationship unclear. Injury in all individuals in intrinsic, susceptible to small fraction of people in idiosyncrasy. Predictable latent period, whereas it is and relationship to exposure to onset of toxicity is variable caused by direct effect of agent and its metabolite, whereas highly influenced by host contribution, that is like immune response, metabolism, polymorphism, etc. Whereas in intrinsic, it is reproducible in animal models, whereas in idiosyncrasy, not reproducible in preclinical animal tests. So, in summary, liver injury classification integrates histopathological patterns reflecting zonal distribution, cellular changes, biochemical markers indicating functional impairment, cell damage, and mechanistic understanding of toxic responses. That is, intrinsic versus idiosyncratic. This multifaceted approach aids in diagnosis, prognosis, and research in liver toxicology and clinical hepatology. So let's look at the final aspect of my presentation, which is initiation, progression, and of hepatotoxicity, how it all happens. It's a complex process, so it's all integrated now step by step on how it all happens. So hepatotoxicity begins with the entry of toxic chemicals into hepatocytes. The main functional cells, the liver's unique anatomical and physiological features, such as its fenestrated sinusoidal epithelium and its role in metabolism, predispose it to injury of various toxicants. So, in xenobiotic agents, through transporters, they enter the hepatocyte, or they enter the hepatocytes, and through because of the SIP, but they get into reactive metabolites. They are metabolized to metabolites, and they are causing this kind of injury. This is the initiation of injury, and the injury can be protein binding, DNA damage, lipid peroxidation, oxidative stress, cytoskeletal alteration, bile duct accumulation, bile acid accumulation, etc. They are also initiation is coming through the intestinal injury infection also. They're also coming from the receptor interaction. So let's see what happens next. Initiation and progression after entry. So chemical entry, lipophilic compounds, including many drugs and environmental pollutants, diffuse readily into hepatocytes due to fenestrated endothelium, allowing close contact between blood and hepatocytes. Sorry, diffuse readily into hepatocytes due to the fenestrated endothelium, allowing the close contact between blood and hepatocytes. Other toxicants are taken up by specific transporters on the sinusoidal membrane of the hepatocytes. What happens in the hepatocytes is bioactivation. Inside hepatocytes, some toxicants act directly by binding to cellular proteins or receptors, while others are bioactivated by enzymes such as cytochrome P450 into reactive toxic metabolites. There are phase 1 enzymes and phase 2 enzymes. Phase 1 oxidative reaction typically generate these reactive metabolites, while phase 2 conjugation reactions usually detoxify them. If toxic metabolite production exceeds detoxification capacity, hepatocytes injury occur. Then there is molecular damage. Reactive metabolites can form free radicals or electrophiles that covalently bind to macromolecules like DNA and proteins, disrupting cellular functions. For example, microcysteines bind to cytoskeletal proteins and causing injury. Next, what happens? Progression. The disrupted hepatocytes homeostasis starts happening in the intracellular signaling, ion imbalance, then mitochondrial damage, caspase activation, nuclear disruption, inhibited repair. So that will all lead to death of the hepatocytes and then liver dysfunction. Then also simultaneously DAMP is produced. So that is activation of endothelial cells, Kupffer cells, stellate cells, platelets, etc. Then leukocyte infiltration, activation of reactive oxygen species, proteases, cytokines, coagulation and complement. All this along with PAMP, which is coming from the gut. So if there is an imbalance here, again death of hepatocytes happen. Cell survival pathways inhibited, then death of hepatocytes happen. So finally it will all lead to liver dysfunction. So again explained here, intracellular events, toxic metabolites formation leads to disruption of hepatocytes, homeostasis including ion imbalance, mitochondrial dysfunction, altered gene expression, and DNA damage. These changes activate cell death signaling pathways such as caspases, leading to apoptosis or necrosis. The release of damage signals. Injured hepatocytes release damage associated molecular patterns that activate pattern recognition receptors like toll-like receptors on non-parenchymal liver cells such as Kupffer cells, sinusoidal endothelial cells, and stellate cells. This activation triggers production of reactive oxygen, nitrogen species, and inflammatory mediators like cytokines, prostanoids, and growth factors. Then there is inflammatory cascade. The inflammatory response recruits leukocytes, platelets to the liver, amplifying injury and promoting repair depending on the context. Activation of coagulation and complement cascades also contributes to the progression or resolution of the liver damage. There is also crosstalk with the gut flora. Disruption of the intestinal barrier can allow pathogen associated molecular patterns, such as lipopolysaccharides from gut bacteria, to enter portal circulation and further stimulate the liver inflammatory and injury. So outcome of this is the balance between injury and repair mechanism determines whether hepatocyte death leads to liver dysfunction, fibrosis, or regeneration. Mitochondria play a central role in deciding cell fate via apoptotic or necrotic pathways, often influenced by the dose and nature of the toxicant. So thank you very much for your patient listening, and you can do further reading Castrate and Dowell Toxicology, the Basic Science of Poisons, 9th edition. Thank you.